So today we will discuss about lipids. So lipids are water insoluble, you know. They could be simple fatty acids. So actually, triglyceride. You must have heard about triglyceride. Triglyceride is made with the glycerol with 3 fatty acids. So lipids, they are having fatty acid. Now fatty acids are what? They are having carboxyl group attached to R group. Now here the confusion comes. R group lipid is also using, and in the amino acid also we studied R group. Is it clear? Here the R group can be hydrogen, it can be methyl, it can be hydroxymethyl. But in case of the lipid, R group can be methyl. There is no, it is not from the hydrogen. It can be methyl, it can be ethyl, or it can be higher number. Means number can range from 1 to 19. So please, this is again important question. R group in the lipid, they can be methyl or ethyl or higher number, means their number can go from 1 to 19. And this is in the fatty acids. Okay, the fatty acids having a carboxyl group attached to the R group. And if they ask you for the protein, then please remember protein in the R group can be hydrogen. In case of glycine, it can be methyl. In case of the alanine, hydroxymethyl. In case of serine. So please remember R group, both they are using it. Here R group number will not be much. It will be either 1 carbon or maximum 2 carbon. But here in case of fatty acid, it can be from 1 carbon to 19 carbon. Now see beta, another book I'll show you. So see carefully beta. This is your, right now we are making the triglyceride. With 1 glycerol, 3 fatty acid condensation is done. Water molecule has gone out. Condensation means water is removed and triglyceride is made. Clear? So now see what we say. Triglyceride is one group of the lipid, and it is example is fat is in adipose tissue in human and oil in sunflower. Now please remember, fat are liquid at body temperature. Body temperature means 37 degree, but fat are solid at room temperature, and ideal room temperature is 20 degree. But oil will remain liquid at both body temperature and room temperature. Now see this in the NCERT beta. See here. So your fatty acids can be esterified with the glycerol. Fatty acids are found esterified with the glycerol. Bond is known as ester bond. And if 1 fatty acid is attached, we say monoglyceride. If 2 fatty acid is attached, diglyceride. 3 fatty acids are attached, triglyceride. And they can be fat or oil based on the melting point. Oil are having low melting point and they remain as a liquid in the winter also. Is it clear? So this is your fatty acids, how they are going for esterification with the glycerol. Now, how many types of fatty acids are there? Fatty acids are classified into 2 categories based on are they having saturated bond only or are they having unsaturated bond. For this, I will show you this diagram. See it. See, this is your fatty acid. One example of fatty acid is palmitic acid. See, it is saturated fatty acid. And if you see the carbon, start from here, number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. 16 is your carboxyl group. So here the question can be asked, how many R group? R group is still 15. R group is having 15 carbon, and 1 is your carboxyl group. Carboxyl group. The total fatty acid is having how many carbon? Fatty acid is having 16 carbon, including carboxyl group. See in our NCERT. Palmitic acid has 16 carbon, including carboxyl group. So palmitic acid is an example of your saturated fatty acid. So how you can write it? CH3, CH2, 14. Okay, because CH3 I wrote it, then this 14, and COH. This is your palmitic acid, which is saturated fatty acid. See in our NCERT, this is the diagram given. CH3, CH2, 14, COH. So this is your R group. This is your R group, and R group is having how many carbon in the palmitic acid? 15 carbon. Please remember, this is the way question can be asked. Is it clear? Now see another fatty acid. Another fatty acid is arachidonic acid. Now arachidonic acid is an example of unsaturated fatty acid. See here, beta. If you see in this case, see the double bond is present. Double bond. So 3 double bonds are there. It can be 1 double bond is there. So beta see one thing, oleic, linoleic, and linolenic, and arachidonic. It is all this classification is done on the basis of how many number of double bonds are present. Oleic is having 1 double bond. See here, through palmitic acid, you are removing one head, you are making a double bond to palmitic oleic. Clear, 1 double bond. Linoleic means 2 double bond. Linolenic means 3 double bonds. See here, linolenic, it is 3 double bond, and arachidonic with 4 double bonds are present. Understood, beta? Now, if you see in the cases in this diagram, all the hydrogen of the double bond are present on one side. See, if they are present on the one side, we call it a cis fatty acid. This is on the one side, means cis fatty acid, and if they are on the opposite side, you say trans fatty acid. So actually, see here, trans fatty acid is not good for health. It is the cis fatty acid which is good for health, because this is the one which will create the bending. Cis will create the bending. Is it clear, beta? Now, when they are having no double bond, you call them as saturated fatty acid. When they are having double bond, then you call unsaturated. Now, if they have only 1 double bond, then you call monounsaturated. If you find more than 1 double bond, then you say polyunsaturated. And all your polyunsaturated fatty acids they are your essential fatty acids. So we say PUFA. PUFA means polyunsaturated fatty acids are EFA, essential fatty acids. See here, omega-3, you must have heard about omega-3 fatty acid. It's an essential fatty acid. So we are saying here also, arachidonic is having 20 carbon, including carboxyl group. So R is having 19 carbon. Fatty acid can be saturated without double bond, or can be unsaturated with 1 or more double bond. Already I've given you example. Now, another type of lipid is your glycerol. Glycerol, if you see, it is also known as trihydroxypropane, because propane is 3 carbon and it is having 3 hydroxyl group. So we say trihydroxypropane. And what we are doing, we are combining now these fatty acid with glycerol to form glycerides. So this is your glycerol. If you are adding 1 fatty acid, CR group, okay, this is your glycerol. To this, you are adding 3 fatty acid. First fatty acid, second fatty acid, third fatty acid, so you are saying triglyceride. Triglyceride. Now, if you see, to make the phospholipid means cell membrane, this was your glycerol, backbone of glycerol, if you remember. This is your 1 fatty acid, this is your second fatty acid, beta. And this is your phosphoric acid. This is phosphoric acid. Clear? And this is your choline. If you remember in the cell membrane, I taught you this is choline. CH2OH, means water molecule has been liberated. And here CH2 and CH3. So choline, phosphoric acid, and glycerol, they become the head. This part becomes as the head. This part. Remember, beta, we discussed this part will behave as a head, and this part will behave like a tail. This is one tail and this is second tail. So please learn this diagram, phosphoric acid, lecithin. Why we say lecithin? Because it is using choline. So what is going on? Some lipids are having phosphorus and they are phosphorylated organic compound. They are known as phospholipid. And very easy example of phospholipid you have to learn is lecithin. Neural membrane are having other types of lipid. We say glycolipid or sphingolipids are there. So this is with respect to the lipid in your NCERT. So I hope so this much topic is clear to you. Let me just explain you how the triglyceride bond is made so that you can remember it properly. This was your glycerol. So this now you are adding fatty acid. R1COOH. R2 COOH. You will liberate the water molecule. This is the way how triglyceride is made. Is it clear? The final product will be what? You can do one thing. We can work here only in this diagram. Remove the water molecule. Okay. And you make a line between them. This is your. And what they have done in the NCERT, they have just made this also as a separate by showing this way. Okay. So you see this with the same diagram as this one. R1 COO to attach to CH2. R2 COO attached to CH. So ester bond is there in the fatty acids. One more type of phospholipid, one more type of lipid is there, that is your sterols, known as cholesterol. In cholesterol, only this is your hydrophilic part and this is your hydrophobic part. Clear, beta? Now 1 minute just. Now see, this is cholesterol. Only this OH part, focus beta, this OH part is only hydrophilic. Remaining is entirely hydrophobic. So cholesterol, it is a type of a lipid, but it is not fat or oil. It belongs to the steroids. Most of the cholesterol molecule is hydrophobic, so it is attracted to the hydrophobic tail of the cell membrane. Only one end is having the hydroxyl group, which is hydrophilic, and it is attached to the phosphate head. That is why, see beta, when you draw the diagram of the cell membrane, you orient the cholesterol this way. Check the orientation of cholesterol, which is helping to maintain the fluidity of the cell membrane. It does not allow the membrane to solidify at low temperature and does not allow the membrane to become too fluid at the high temperature because of its orientation. That if you remember, I focused it also when I explaining you cell membrane. This is the way how orientation of the cholesterol will be helping in the packing of the phospholipid bilayer. Up till here, clear, beta? Any doubt? We'll start with the carbohydrate. First of all, carbohydrate, you know, they are hydrates of carbon. Hydrate means water molecule of carbon. So whatever it is present carbon, same will be the number of water molecule. If suppose I substitute N value with the 6, then you are writing C6H12O6. So here hydrogen to oxygen ratio is always following the water ratio 2:1. Now carbohydrates, they are of different categories. They are monosaccharide. Depending upon how many carbon, if they are of 3 carbon, you say trio sugar. Trio sugar example is your glyceraldehyde. If it is of 4 carbon, you say tetrose sugar. Example is your erythrose. 5 carbon, you say pentose sugar. And you will say ribose or ribulose. 6 carbon, hexose sugar. Example, glucose. Galactose, fructose. Aldehydes are present as a functional group or ketone is present as the functional group. So if aldehyde is present, example of triose is glyceraldehyde. In 5 carbon, it is ribose. And in 6 carbon, it is glucose and galactose. They are the one which are having functional group as an aldehyde. But if suppose functional group is ketone group, then we will say ribulose. If you remember in the photosynthetic chapter, we say first carbon dioxide acceptor of C3 cycle is a ketose sugar because it is having functional group ketone. Fructose is also ketone. In the trioglyceraldehyde, you will see another ketone sugar is present, dihydroxy acetone. And all this you have studied in the glycolysis. So these are the example of your ketone sugar. Now when two monosaccharides, they are combining, so they are becoming as disaccharide. And example is lactose, which is made by the glucose and galactose. So lactose is a product of glucose and galactose. Sucrose is a product of glucose plus fructose. Glucose plus fructose. Maltose is a product of glucose plus glucose. So when you are combining them to make disaccharide, obviously condensation reaction will be there, means water molecule will be liberated. And the bond present between them, the bond is glycosidic bond. So glycosidic bond will be present in between them. Is it clear? So monosaccharides are joined to form disaccharide or polysaccharide with the help of glycosidic bond. Now let me show you the NCERT beta. 1 minute, just a sec. See here. See the line carefully. In the polysaccharide, the individual monosaccharide are linked by glycosidic bond, which is made by dehydration. All the bonds are made by dehydration, means removal of the water molecule. This bond is made between the two carbon atom of two adjacent monosaccharide. So this much I hope you have understood. Now let us see the example of polysaccharide. Now when you see of the polysaccharide, they are of three categories: starch, cellulose, glycogen. First of all are homopolymer of glucose. They are homopolymer of glucose. This is also homopolymer of glucose. This is also homopolymer of glucose. Next is your inulin. Inulin is a homopolymer of fructose. And last is your chitin. Chitin is also homopolymer of modified polysaccharide. Modified polysaccharide example is your N-acetyl galactosamine. Now one thing beta, question can come in the mind. Plant is storing the starch, it is making the cell wall of cellulose, both are polymer of glucose. Then what is the basic difference between them? Basic difference between them is starch is making use of alpha glucose. Let me explain. Starch is making use of alpha glucose and cellulose is making use of beta glucose. It is made with the alpha glucose and cellulose is made with the beta glucose. As a result, if you know beta, starch will make the helix. This helical form of the starch is known as amylose. And at one or two places, branching will be there very less. Branching very less. So amylopectin. Helical form is amylose. And this branching form with a few helix area, that is known as amylopectin. So starch is having two things, amylose and amylopectin. Now, if you see in case of the animals, they are also making use of alpha glucose. And see carefully the diagram. Helix will be there, but very less helix and branching will be more. More branching will be there. Extensively branching, it means it is more in the amylopectin form. If you remember, beta, we discussed this term, amylopectin, we discussed in the red algae. In the red algae also we told that red algae is storing fluoride and starch similar to amylopectin and glycogen in structure. So glycogen means it is having more extensively branched and starch is actually less extensively and more is amylose. Learn this way. If you remember beta, enzyme which break down the carbohydrate, we gave the name to the enzyme is amylase because it is going to break amylose. Now come to the cellulose. In case of cellulose, you will not find helix, you will find only linear strands. Cellulose microfibril will be there, which are stabilized by the hydrogen bond. Hydrogen bond will be present in between them. So they are not making helix And when they are not making helix and you want to do the starch test, it will come negative. I think starch test, it will come negative. I think so this much you are understanding it. Now see the NCERT diagram carefully, beta. See, this is the diagram of glycogen. And in the glycogen, if you see, first of all, bond is, check, this is your first carbon. Okay, I'll focus here. First carbon, second, third, fourth, fifth, and sixth. Once again, 1, 2, 3, 4, 5, and 6. So I think so you can see a bond is made between first and fourth carbon. Between first and fourth. And if you see here, OH was down. Here OH was down. That is why this bond was made. And here also OH was down. So this is alpha. This is your O. Next to the O carbon, if OH is down, you say alpha. If OH is up, you say beta. See one more diagram. Try to understand better. See here, this one. Next to the O, the first carbon, if OH is down, you say alpha. And if I draw OH upside, instead of this, let me assume if I'm drawing it this way, what you will say? This is beta. Clear, beta? If OH is down, it is alpha. So this is your alpha glucose. In the glucose, 6 carbon are present, but ring is having only 5 carbon and fifth is holding the 6th one. This is the example of the 5 carbon ribose. It is also aldehyde. In the ring, it is having 4 carbon, 1, 2, 3, 4, and fourth is holding the 5th carbon. So to make starch, in the diagram, I think so I tried to explain you, we are using glycosidic linkage between 1 to 4 position, and we are using alpha glucose. Now you know this is your 6th carbon. You know this very well. And this is your first carbon. Now see branching. Branching is occurring at a point between, and the bond is made between the first and the 6th. 6th carbon of the linear chain, 6th carbon of the linear chain attached with the first carbon of the branched, first branch. Is it clear? So whenever this branch point is coming here, so this linear, now it is linear line for this branch. So linear 6th carbon and branching first carbon will be used. So see beta, diagram is telling you glycogen is having so much branching. See, it is similar to amylopectin. And I told you cellulose will not make helix. Cellulose does not form helix, and that's why it cannot hold iodine. But starch is able to form a helix, I've told you, secondary structure. And it is able to hold iodine. That is why it gives blue-black color. Up till here, clear beta? So what are acid insoluble pool is having? It is having polysaccharide, which is actually nothing. It's a long chain of the sugar. We have already studied that starch is what? Starch is the one which is used for the storage in the plant tissue. Animals are using for storage glycogen. Plants are using starch. Plants are not only using starch as a storage, they are making use of the polysaccharide to prepare cell wall. Cellulose is a polymer consisting of only one type of monosaccharide, that is glucose, and it is homopolymer. Starch is also homopolymer, cellulose is also homopolymer, glycogen is also homopolymer. Up till here, clear beta? Now when, see one thing beta, sugar, if you know, there are 2 types of sugars are there, reducing sugar and non-reducing sugar. Reducing sugars are those sugars which are able to donate their electron, which will donate the electron and make the other person reduce. Is it clear? Benedict test, if you remember the test we discussed also, test for presence of glucose is Benedict test, in which it is generally done to know the people is suffering from diabetes or not. If the people is suffering from diabetes and diabetes is crossed the threshold limit, it will come out in the urine, and when you take the urine and do the Benedict test, it will turn the copper Cu2 plus into Cu plus. Okay, so color will come, red color, precipitate will come. It means it can give the electron. So we say it is a reducing sugar. So in case of the polysaccharide, right end is reducing end and left end is non-reducing end. In case of polysaccharide, please remember all monosaccharide are reducing sugar. All monosaccharide are reducing sugar and disaccharide are reducing sugar. And in polysaccharide, Right end is reducing, left end is non-reducing. Only one example, sucrose is a disaccharide which is non-reducing sugar. Sucrose is a disaccharide which is non-reducing sugar. Why? Because it is made with the glucose and fructose, aldehyde and ketone. So all the functional groups are used. That is why we say sucrose is non-reducing sugar. So I hope so up till your concept is clear. Now see, you know very well plant cell wall is made up of cellulose and paper is made from the plant pulp and cotton fiber is nothing, cellulose only. So this much is clear to you. Now, apart from this, there are other more complex polysaccharide present and they are building blocks as an amino sugar and they are chemically modified sugar, for example, glucosamine. It means you are adding amine group to the sugar glucosamine or N-acetyl galactosamine. So they are your modified sugar, or you can say they are complex polysaccharide. Example is chitin, and chitin is your homopolymer. Chitin we see in the fungal cell wall, we see chitin in the exoskeleton of arthropoda also. So I hope so. Today in this up till here lipid and carbohydrate according to the NCERT we have covered.